So hello students, this is the next class. At last class we studied about RTK and in this class we'll study about the NRTK, non-receptor tyrosine kinase. What is this? It's a cytosolic enzyme that transports phosphate group from ATP to tyrosine residue on target protein. This normally transfers the phosphate group from the receptor and C-terminal to the tyrosine residue of the target protein. In RTK, it have an intrinsic enzymatic kinase domain bound in the receptor that receives the phosphate group and activate further. But in NRTK, the receptor not having any intrinsic enzymatic activities, the enzyme non-covalently associate to the receptor cytoplasmic domain. For example, SRC family kinase, JAK, that is genus kinase, STAT, etc. Activation mechanism: ligand bind to the receptor, lack intrinsic kinase activity. Here, receptor dimerized. After the dimerization occur, the receptor recruits some NRTK, that is JAK or STAT. And this JAK or STAT cross phosphorylate and means the receptor cross phosphorylate and activate the JAK protein and the further downstream target activated. Let's see the JAK state pathway here. In the in the receptor on the extracellular side, that is cytokine receptors, cytosine binds and dimerize the receptor. After dimerization, it associates the JAK protein. The JAK protein bind to the C terminal, and after cross phosphorylation, they get activated. Due to activation of JAK, uh, further docking site is open for STAT protein. STAT protein bind and they get activated from the JAK protein. After the activation of STAT, that is through the phosphate groups that has been sent from JAK to activate the STAT. After the activation of this state, they got dimerized and travel to the nucleus through the nucleus pores. After the after after the entering to the nucleus, it bind to the DNA and initiate the transcription or transcription of immune and growth genes. So talk about next, that is TGF beta receptor pathway. Here the receptor activated, how the receptor activation occur. TGF beta bind to type 2 receptor, which then recruit the type 1 receptor and the cross phosphorylation occur. After the cross phosphorylations, they recruit SMAD2 and 3 and phosphorate them too. The SMAD2 and 3 unwind from the receptor and co-associated with the SMAD4 and enters the nucleus, and the expression of gene occur. Then let's talk about the dual role of TGF-beta in cancer. Here, the first, in the early stage, tumor suppressor, it acts as tumor suppressor. Here, in healthy cell and early stage tumor, TGF-beta induce cell cycle arrest and promote apoptosis to neutralize the cell concentrations and growth, act as a natural brake and cellular proliferations. The second, later stage tumor that is tumor promoter, cancer cell mutate to evade TGF-beta induced apoptosis. Higher level of TGF-beta in the tumor microenvironment suppress the immune system, inhibit T cells. It inhibits T cells and act on TGF-beta heavily driven EMT, that is epithelial mesenchymal transitions, transport stationary epithelial cell into high mobile mesenchymal cell, promoting tumor emissions, drug resistance, and metastasis. Let's talk about receptor guanylyl cyclase. Here, it is a single pass transmembrane receptors with an extracellular ligand binding domain and an intracellular guanyl cyclase catalytic domain. Here, the ligand responds to the neuritic peptides, that is ANP and BNP. After the binding of ANP and BNP, uh, after the binding of ANP and BNP, the conformational change and intracellular catalytic domain activate. This uh, takes 2-phosphate from GTP and forms CGMP. After the CGMP form, it acts as a secondary messenger and activates protein kinase G that regulates ion channel and smooth muscles. Let's talk about atrial natriuretic peptide ANP pathway. Here, the heart arteria stretch due to high blood volume, and due to that, ANP secretes. ANP bind to the receptors, guanylyl cyclase, and uh, that present on smooth muscles or kidney cells that stimulate GDP to form CGMP, and this CGMP activate PKG, protein kinase G, and this protein kinase G act on calcium channel and also on smooth muscles relaxation. During the activation act on calcium channel, it stops the calcium to uh, to travel inside the cell, and uh, dephosphorylations of MLC, that is myosin light channel, result in relaxation of vascular smooth muscles. Due to this, the physiological effect cause powerful vasodilations, increased sodium excretions by kidney, overall reductions in the blood pressure. Then talk about inotropins, that is CAMP or calcium mechanisms. Here, hormones that alter the force of muscles contractions, especially heart muscles. Two types, that is increased contractility and decreased contractility. Increased contractility example epinephrine and uh, duotamine, and decreased contractility beta blocker and calcium channel blocker. Mechanisms are when epinephrine bind to beta 1 androgenic receptor, that is GPCR. Here the G alpha S activate from through the through GEF that change GDP to GTP, and due to that the adenyl cyclase activate. Sorry, G alpha S activate the adenyl cyclase. After the adenyl cyclase activate, it stimulate the ATP to form CMP, and that CMP will activate the PKA, protein kinase A, and that protein kinase A activate the L-type calcium channel. Due to that, the calcium enter to the cell and increase the calcium uh, level. Due to the increase in intracellular calcium level, it triggers strong actin-myosin cross-binding. Here, clinical evidence crucial in managing heart failure and cardiogenic shock and several hypotensions. So that's all for today. We'll talk and another part will I will upload tomorrow. For now, this all. Have a nice day. Thank you.